All right, hi again, everyone. We're on to lesson 2, the cell cycle, cell division, and cancer. Because this is just the beginning of the course, I want to make sure that everyone's on the right page. So let's just quickly take a look at Brightspace here. So at this point, you should be finished lesson 1. So you should have done all these things. You should have downloaded your glossary. You should have watched my first video. And if you wanted to use the slides, they would have been posted there. You can't see them right now, but they will be there by the time that you're reading this. You would have used this website to help you fill in the glossary. You would have completed the first quiz on organelles, which is that multiple choice quiz. So if you haven't done all those things, please go back and do them. If you have done all those things, then let's continue on with lesson 2. So what you're going to have to do in lesson 2 is, if we click on lesson 2 here, watch this video, complete more of your glossary from cell cycle up to and including chemotherapy, complete some questions from your textbook. So I have the textbook pages you need right here. You will click that and go to page 39 and complete questions 1 to 7. So if we click that, then you go down to page 39, which is the last page, and complete questions 1 to 7. You can skip question 8, and then you can see that you have an assignment to do after that. Okay, so this is everything that is needed in lesson 2. So let's get on to it. All right, learning goals. What you're going to have to do in this lesson is describe what happens during the cell cycle. Describe how cell division occurs in animals and animals and plant cells, and explain the connection between cancer and cell division. So we're going to talk a lot about how cell division happens and connect that to this idea of cancer. Okay, so this is the cell cycle. What this is, if you like, is sort of the life of a cell which repeats. So you can think about it almost like our lives, right? You get up in the morning, maybe you eat. I hope you eat in the morning early on. You go about your day, you go back to sleep, you get up the next morning, and it just continues. So this is basically a cell's life, so to speak. So let's start here at this point. So after a cell divides and you get 2 new cells out of that division, both of these cells are going to go through this process. So they're usually going to go through what we call G1 phase. So they've just divided. They basically came from one cell that split into 2, so now they're both quite small compared to the cell that they came from, and they're missing some of the organelles, right, because they had to split the organelles between them. So what they have to do is grow. So G1 stands for gap 1, but you can think of it as growth, right? So cellular contents, excluding the chromosomes, are duplicated. So they're going to start making new organelles, right? They're going to duplicate those so that the cell can grow. Gap 0 over here, we're not going to worry about so much, but basically a cell can kind of freeze, if you like, and stop going through this cycle. But generally speaking, we're going to go through here. When we get to this point at the end of the gap 1, there's a cellular checkpoint where a cell checks to make sure that everything's okay. If everything is in fact okay, then you go into S phase. And in S phase, S stands, you can think for as synthesis. What's going to happen is the cell is going to copy its genetic information. So all that DNA that is kept inside the nucleus is going to be duplicated so that you now have 2 copies of your DNA, and that DNA is wound around proteins, and it forms structures called chromosomes. Now this is a little bit misleading. You're not going to really necessarily see those chromosomes, but they are duplicated. You now have 2 sets of genetic information here, whereas here you just had 1 copy of it. You now go into G2, where the cell double checks that everything is okay, and if it is, then it can move on to mitosis, and it will divide again to form 2 new cells. So mitosis is the process of cell division. Well, I should say that's not quite right. Mitosis and cytokinesis are the process of cell division, and we're going to talk about those a little bit more. G1, S, and G2, and G0, I guess if you like, all make up what we call interphase. So this is sort of the regular life of a cell when it's not dividing. It's just doing what a cell does. And then this part here is cell division. Okay, so cell division. What happens in cell division? Well, we have 2 main components. We have mitosis, which explains how the nucleus splits apart, and then how you get 1 nucleus to form 2. And cytokinesis, how the rest of the cell splits apart and how you ultimately get 2 cells derived from 1. The other thing to note is that these cells are clones of each other, right? This is not like sort of like human reproduction, sexual reproduction, if you like, where you end up with offspring that are a combination of two different parents, mitosis produces clones. So the two cells that come out of this process are both genetically identical to each other and genetically identical to the original cell that split apart. Okay, so how does mitosis happen? So we're going to go through this at a relatively basic level. So as long as you have a general idea of what happens in each stage, you'll be fine if you know it sort of to this level here. So what we start to have happen is you can see here that this is basically, you're starting to get into mitosis, but you're still kind of like right at the end of interphase. You can see that these chromosomes are not kind of forming those classical X shapes, right? They're kind of like unwound. And that's basically because if you remember, the nucleus controls the cell. It's got DNA on it, and that DNA provides instructions for the rest of the cell, telling it what to do. If the DNA is unwound like this, then those instructions can be accessed. It's easier for the rest of the cell to understand the instructions. If it's condensed like this, it's harder for the rest of the cells to use. If you want a sort of analogy here, think about the stuff in your room, right? It's probably relatively spread around. I mean, even if you're an organized person, you're going to have different drawers with different things in them. So generally speaking, in your day-to-day life, your room probably has things like this. But what if you have to move, right? You're moving to a new home. Well, then you got to pack everything together. That's going to look more like this. This is a convenient sort of arrangement for when you're moving, to pack everything together in one place, right, in a box, so it's easier to carry. But it's hard to access, right? If you want to, I don't know, whatever you have in that box, if you want to play video games, if you want to read a book or whatever, it's harder to get at it if it's the bottom of a box somewhere. So this arrangement is great for sort of everyday use, but it's not good for moving. This is good for moving, but not necessarily the best for everyday use. So when you see chromosomes like this, that tells you, okay, it's moving time, right? The cell's going to divide. So remember that when you get to this point here, which is the beginning of cell division, this is prophase, you already have gone through S phase. So you already have 2 copies of your genetic information here. Okay, so what's going to happen is that genetic information is going to condense into these chromosome structures, and it'll become visible under a microscope. You'll be able to see this. The nuclear envelope is going to start to break down. Remember that there's a membrane. It's in fact a double membrane around the chromosomes here. So the nucleus is surrounded by a membrane. That's going to break into pieces. And if it's an animal cell, you're going to start to see these things moving to the poles of the cell. These are your centrioles. And what they're going to do is produce spindle fibers, which, if you remember before, we talked about cytoskeletons, and it's a little bit like Spider-Man shooting threads around. That's kind of what's going on here. And those threads are going to be what is going to grab onto the chromosomes and pull them apart. So by the time you get towards the end of prophase, you have the centrioles have gone to the poles, sort of the outer positions of the cell. The spindles are going to be latching onto the chromosomes, and their chromosomes are condensed. So if you want, we can basically break this down into 3 things that are going on here, just to keep it simple. 3 things, okay? Centrioles have moved to the poles and are producing their spindle fibers. Chromosomes have condensed and are preparing to move. Nuclear envelope has broken down. That's it. This is prophase. Next, we move on to this phase, metaphase, where we can see that all the chromosomes have lined up in single file along the middle here, the metaphase plate, right? They're right in the middle, and you can see the spindle fibers are now attaching to the center of the chromosomes there. If you've ever heard of the term kinetochore, don't worry about it if you haven't. They're basically they're connecting to the center up here. And what's going to happen is those spindle fibers are going to contract, and they're going to pull those chromosomes apart. But in metaphase, everything is just lined up in the middle, and you can see how this is going to work, right? Because if you think of this red chromosome here, we said that the DNA has been duplicated. There are 2 copies. Here's 1 copy on the left. Here's 1 copy on the right. If you pull them apart, 1 copy goes one way, 1 copy goes the other way. If everything is single file, then every single side is going to get 1 copy of 2. So if you imagine in a human being, right, you might know that a human being has 46 chromosomes. You would see 46 chromosomes line up like this. And because there are duplicates, 1 equal portion of that 46 chromosome goes this way, the other goes the other way. and so you have equal amounts of information here and here. So you have two equal cells. Okay, here we have anaphase. Anaphase is where the chromosomes are now actually being pulled apart. Here they're just kind of lining up in the middle and preparing to be pulled apart. Here now they are being pulled apart. So they're moving towards opposite sides of the poles. You can also see that the cell is starting to elongate. It's starting to get longer to accommodate this movement. Okay, so this is still anaphase here. So the chromosomes are being pulled apart, the cell elongates, and you're starting to get some pinching in the middle here, preparing for the cell to separate. In telophase, you can see that the chromosomes have fully moved from one side to the other. You're getting nuclear envelopes reforming, so you're forming two nuclei, here and here, and the pinching inward has continued. So we sometimes say cytokinesis happens after mitosis, but really what you're seeing here is the beginnings of cytokinesis, the breaking apart of the cytoplasm. So that actually starts to happen at the end of mitosis. Finally, at this point, we have had cytokinesis happen. So the two cells have broken apart entirely. It's not just the nuclei that have duplicated, but now the two cells have split, and now we're ready to go into gap 1. Okay, so that's the process of mitosis here. This is just to show you really quickly, cytokinesis happens a little bit differently if you're talking about an animal cell or a plant cell. So in an animal cell, we have this pinching in that happens. We call that a cleavage furrow. And so you have a ring of some of the same material that you have in the cytoskeleton, which contracts and breaks these two things apart. In a plant cell, it's a little bit different. You have a structure forming in between where the two cells are going to separate, and you actually have cell wall build there in the middle, right? Remember that plant cells have cell walls, animal cells don't, so you actually have a cell wall form in between the two cells, and they separate that way. And we call this region where this formation is happening a cell plate. Okay? So my question for you is, if we have interphase, which is composed of gap 1, S, and gap 2, and we have mitosis, which is composed of prophase, metaphase, anaphase, telophase, plus cytokinesis at the end, where does a cell spend most of its time? So I could just tell you the answer to that, and I guess I will, but it's kind of cool because you can kind of figure it out by looking at pictures like this. So what this is a picture of a root of a plant, and this is an area of heavy cell division. And you can imagine why that would happen, right? If you have the tip of a root, the root is growing into the soil, it's getting longer, so if you're having growth, you're going to be having cell division. So this is an area of very active cell division. So we can look at the cells and just ask ourselves, okay, we have very active cell division here. What phase are most of these cells in? And if you look at this, you can see this looks maybe like it's a metaphase of some kind because we have the chromosomes lined up in the middle. Here we might have a prophase because you can see the chromosomes are starting to condense. But you've got a lot of these things where you don't really see the chromosomes. You see a fully formed nucleus, and that's it. So what that tells you is the cell actually spends most of its time in interphase. And that makes sense when you think about it, right? You don't spend most of your time moving. You spend most of your time at your home doing the things that you need to do in life. And that's going to be true for a cell as well. Okay, so now that we've talked about the cell cycle and cell division, we're going to look at how this relates to the topic of cancer, which is a really important topic for us. It's quite a scary disease. I'm sure all of us probably know somebody who's been affected by this disease. So this is a very serious concern for us. So how does cancer, the disease, relate to everything that we're talking about here? Well, cell division, as we've just described it, right, meaning the cell cycle followed by mitosis and cytokinesis, is a highly ordered process with many checks and balances. We said that a cell actually checks what's going on with itself. We're not going to really get into how, but it checks it at the end of G1 and checks it during G2 as well. So it's highly ordered. The cell tries to make sure it's doing everything right and has many checks and balances to ensure that when it's dividing. In fact, cells can generally only divide about 50 times before they're no longer able to. There's a reason for that we're not going to really be able to get into, but it's thought that part of the reason for that might be to prevent cancers from happening. So there's a limit. You can divide about 50 times, and that's it. And that might actually have something to do with people think that maybe there's a limit to how old people can get, right? Like with advances in medicine, you can improve lifespans up to a certain point. You have more people living longer, but you still don't have people living to 130 years old, right? There may be a limit to how long you can live based on your cells being able to replace themselves. Just kind of an interesting thing. If you want to look up telomeres or Hayflick limit, you could find out more about this, but I'm not going to test you on that. So anyways, cell division is highly ordered, and cells are only able to divide about 50 times. If a problem is noticed by a cell where it notices there's something going wrong with its cell division process and it can't fix the problem, then the cell will generally kill itself. It'll commit suicide by a process called apoptosis, which is also a highly ordered process. So the cell doesn't just like shred itself and die. It carefully wraps up its different components and has a really organized cell death. And that's important because some of the components of that cell can be quite harmful if they're allowed to leak out into the environment. So that's apoptosis. So why would a cell do this? Why would a cell be so concerned that its division process be well ordered? Well, let's get into that. The process of cell division is controlled by the nucleus of a cell, which has DNA in it, right? And the DNA provides the instructions for a cell, telling it essentially what to do. So ribosomes and other organelles will build proteins based on the instructions in the nucleus, right? The instructions that are written in the language of DNA. These instructions can become corrupted, or in other words, they can be damaged so that there's a mistake, right? DNA is like a code, and if you break apart a code, then it can say something else, right? Just like if you have a sentence in English and you take out a letter, its meaning can change. So the same thing is true of DNA. And so ways that DNA can be damaged: UV radiation from sunlight, for example, certain chemicals can do it, or even just copying errors. So just sort of the daily life of a cell produces enough chaos that this stuff can just happen sometimes. And so the DNA instructions are corrupted, and they no longer say what they originally said. And this is a problem because we said that cell division is a highly ordered process. So if the instructions are corrupted, then the order of that process can break down. And that can cause a cell to divide uncontrollably, and it may be able to divide more than 50 times. So the cell will just keep dividing and dividing and dividing and creating more and more cells. And so when it does that, you can get a tumor or a mass of cells. Okay, so basically what you have happening here is cell division is an ordered process because the instructions that essentially tell it to happen become corrupted. Okay, so a tumor can form, but that doesn't mean that if that happens that a person has cancer, because not every tumor is cancerous. Some are what we call benign. They can still be quite harmful, or in some cases not harmful, but the actual cell division process is limited. The cells will divide for a while and then they're going to stop. And that might be because if you can imagine these cells expanding outwards, there's a blood supply in there. And if the cells move away from the blood supply, they don't get oxygen, they can't create the energy that they need to live. And so any cell beyond this part of the tumor might just die. And so the cells are limited to this area here. So that would be a benign tumor. But some cells don't only lose their ability to control their cell division, but other issues come up as well. So for example, some cells can actually cause the growth of blood vessels to happen near them, which sounds really creepy and scary. And how does that even happen? But there are, when we talk about cell specialization, it might sort of make a little bit more sense. But cells do have instructions for providing blood vessels into a certain region. And you can imagine we might need those when we're developing as embryos, right? If you're developing from a ball of cells into a baby, you're going to have to grow blood vessels to support those cells. And so some of the genes that actually control our development from an embryo up to a baby, some of those genes get corrupted when we have cancer happen. Now, I say genes. What I mean by genes is the sections of DNA that provide instructions. Okay. So in the cases of some tumors, the cells actually have the ability to not just continue to divide, but to do other things that allow them to spread or to invade tissues. So they're able to move around beyond just where they're at. And we call that metastasis, and we say that those cells are metastasizing, and that's bad because these things can now spread throughout the body, and you can have tumors popping up in multiple places. And they can put pressure on vital organs. They can block the important ducts that carry materials from place to place. So this is why cancer can be so dangerous. Okay, so if that's how it happens, how do we treat it? Cancer is hard to treat for a number of reasons. I'm going to focus on two of them here. And that's because cancer cells are a lot like normally functioning cells, right? They have the same organelles that most of our cells have because they are basically our own cells. They've got some damage in them, but they are still otherwise like our cells. So how can you actually attack them with drugs without killing the normal functioning cells in our body? It turns out that that's really hard. And that's why some cancer treatments have severe side effects. Like if you think about chemotherapy, you might know that people that have chemotherapy can feel quite sick. They can feel quite tired. They can lose their hair. It's because these drugs are harsh on the body's normal cells in addition to cancer cells. But we're just hoping that the cancer cells are hurt that much more. Right? And then you have things like radiation as well, which you can basically aim at a tumor of cells. So you're not hitting all of the cells in the body. You can localize your treatment. But that's a bit tricky if the cancer has spread, right? So it's hard to treat. The other reason it's hard to treat is because a cell can become damaged in so many different ways that could cause it to become cancerous. Cell division is a highly ordered process, which means there are many, many instructions involved in regulating it. And those instructions can go wrong for different reasons. And so all of those different reasons are going to be different cancers. And so cancer is not one disease. It's many, many diseases, and we can't treat them all in the same way. So it's tricky, right? You have to kill something that's very similar to basically other cells in our body. And there are so many different ways that it can happen that if you come up with one treatment that's effective against one type of cancer, there are many other cancers where that treatment might be less effective. Okay, so cancer treatments tend, as we said, to target localized areas. So you might take radiation and bombard an area where there's a tumor. You might extract that tumor through surgery. And if you want to go for basically treatments that target more of the body, then that's where you'll take something like chemotherapy, but that can be quite harsh on a body. Okay, so that's basically all I wanted to say about that. I just added a couple little extra things in here if you're interested that I think are kind of cool. If you've ever heard of this story, this is a really interesting story. This woman here died of cancer in the 1950s. I think it was cervical cancer that she had. So I wrote that here, so that's I think I looked that up, and that's right. So what happened was when she was being treated for cancer, they had a biopsy, meaning they took some of her cells to examine them, and they found that those cells were able to grow in a lab much better than many other cancer cells. And so they were able to study these cells. So even after she died, her cells were still growing in a laboratory. And these cells have actually been used. You can infect them with viruses. You could do things to them, do research. So vaccines were produced based on these cells. I think even the polio vaccine was created in part from these cells. You can do cancer research on these cells. And so her cells are actually still alive. They're used all over the world, and they're even a bit of a nuisance in the laboratory because they're so good at growing that they can contaminate other experiments. But her cells were taken without her permission. And it was only in 2013 that her family and the NIH, the National Institute of Health, came to an agreement on how these things can be used. So her name was Henrietta Lacks. It's an interesting story. You can look it up. There's a lot of material on this story now. And the last thing here, I'm not even going to talk about this, but if you're interested, there is a really kind of scary example of a cancer that can be contagious. Fortunately, this isn't in humans, it's only in dogs, but it's kind of similar to this case in the sense that you have cancer that's able to survive beyond the host's body, but instead of surviving in a lab, it can actually survive in another dog. So it's sexually transmitted. So if one dog infects another dog, the cancer can jump to that dog and then keep moving from one dog to another. And they've studied this cancer, and they figured it actually developed in the dog thousands of years ago, and is being passed on. So just if you're interested in this stuff, it is pretty fascinating, while it's scary at the same time. Okay, so that's all I've got to say for this lesson. Just one more. Have a good one.